இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Thank you, Harishweta. Ma'am, shall I proceed, ma'am?
Yes, ma'am.
It takes vision and courage to create. It takes faith and courage to prove. Now, I would like to invite our Honorable Principal, Dr.
Hello.
Is it audible?
Is audible?
Yes, sir. Yes, sir.
Very good morning to all present here. It is a great day to see the most eminent personalities in a virtual platform. I am very happy to welcome you all to this, the International Conference on Sustainable Development Goals in Biosciences, organized by the Department of Biosciences, Vivekananda College of Arts and Science for Women Autonomous, Vivekananda Arts and Science College, Sangagari. This conference is specially meant to update the latest development goals in biosciences. It provides an ideal platform and opportunity to share research ideas with eminent scientists, academicians, scholars, students, and industrialists working in various fields of the advanced sciences. Review challenges have been faced by the community in the present situation, and the scientific deliberation will be in the form of the invited talks, the oral and poster presentation. For these few words, it is my bound duty to welcome you all for this second international conference. As the principal of the esteemed institution, I first of all thank our beloved chairman, sir, who has to shower with us his generosity, and he is always noted for his vision on the women empowerment and industrious workmanship on the welfare of society. That's why we have such a biggest institution with the potential in an unindividual village. I must thank him not only for the opportunity, but on behalf of the person with a social consciousness, I proudly welcome you, sir. I welcome our chairman, madam. Though she is in absence here, yes, we know there is a woman beyond every successful man. She is there. They send us her blessings and wishes for the great success for this event. She is the lady who walks along with our chairman, sir, and leads us to follow the head. And not only for this official, but also the personality. I proudly welcome you, ma'am. On behalf of our institution, I am very proud to welcome our chief guest, Dr. Arun Mayalagan, the Chief Director, Cytogenetics, Department of Aerospace, Biomedical Research Center, Institute of Medical Sciences and Genomic Medicine, Coronia, Spain. Institute of the Busy Studio, sir has accepted the invitation. So being with the chief guest, sir is going to deliver the inaugural address in this second international conference. On behalf of each and every one of them, I extend you warm welcome, you, sir. I have no doubt that there is a vision and sense of the purpose which forms the building block of our institution. Their enthusiasm and contagious and cherish it. I use the golden opportunity to welcome our trust people who have been contributing the peerless effort to the development of our institution. We miss among us for our most respected, our Joint Managing Director, Dr. Athanadiswaran, sir, the most respected, our Joint Secretary, Dr. K. Sri Raganidhi Arthanswari, madam, the most respected, our Vice Chairman, Dr. K. Kribanidhi, sir, the most respected, our Director, Dr. Nivedana Kribanidhi, ma'am, who are all very supportive for this international conference, who are unable to present here with us today due to the another engagement. I am very happy to extend you all a sincere welcome you all. Next, I extend a warm welcome our Executive Director, Dr. S. Kuppusamy, sir, the man who guided us to perform the development of our institution. On behalf of each and every one of them, I extend you a warm welcome you, sir. I use the great opportunity to welcome the most respected, the Director of Academic of Arts and Science Colleges, Vivekananda College of Vivekananda Educational Institution, Dr. Kumaravel, who is continuously his knowledge and experience efforts to building up the institution to remarkable status. Our chairman, sir, Vision, I welcome you, sir. Next, I extend a warm welcome, Dr. N. Rajendran, the principal of Vivekananda Arts and Science College, Sangagiri. His presence in this international conference, I welcome you, sir. I extend a warm welcome over the Dean of Biosciences, Dr. T. Shankar. He is the one who contributing his efforts and his knowledge for the progress of our each and every step of our institution. On behalf of each and every one of them, I extend you a warm welcome you, sir. On behalf of such institution, I welcome you abroad, our institution to glorify itself by our valuable presence and presentation. I assure this opportunity is one of the remarkable milestones of our institution history, where the experts who came from all sides of the India with a prolonged education and knowledge history to make us a step ahead. I welcome you all once again for accepting an invitation and present here. So we have the heads of the Department of the Biosciences of Vigas and VCW, and convener and an organizing secretary, who are all the real backbone of these departments. Due to this earnest effort, the college could grow to present stature. It's my proud privilege to welcome you all. Last, not but the least, okay, to forget the welcome our princes of occasion, who are the bona fide on this international conference. I shower my welcome all of you, dear students of our institution, and as well as the guest institution. I also take this opportunity to welcome the memorable faculty members and staff members and other guests present here. Once again, I welcome you all for this second international conference related to the biosciences. Thank you very much for giving me an opportunity to welcoming the gathering. Thank you, sir.
Thank you, sir. Practical, realistic, and the language of visionary and the idealist. None other than I would like to call upon Professor S. Kuppusamy, sir, Executive Director, Vivekananda Educational Institutions and Hospitals, to deliver the felicitation address.
Yeah. Good morning. Am I audible?
Yes,
Yes,
sir.
sir. Audible, sir.
Okay, thank you.
Yes, sir. Audible, sir.
The principal of the Vivekananda College of Arts and Science for Women, Autonomous, Dr. Suresh Kumar, the director of the Arts and Science Colleges, Professor Kumaravel, Dr. Rajendran, principal of the Vivekananda Arts and Science College for Women, Sangagiri, the deans, heads of departments, faculty members, the participants of this second international conference on sustainable development goal in biosciences, the students, the chief guest of the today, who has agreed to talk about his own experiences on the topic, and other speakers, ladies and gentlemen. Again, it's a good morning to every one of you. It's indeed a pleasure for me to get associated with this second international conference that is being organized on a topic which is highly relevant with the today's context. You all know that we are all living in the context of fear and uncertainty in every sphere of life, not only in this part of the country, almost in the entire country, and of course the entire world. The whole world is almost has come to standstill since last January or February 2020, and it continues. We are calling it wave 2, and in the wave 2 things are going much bad. And why this is happening? And that is where I see the conference theme is just coming in to the reality. See, when you talk of a sustainable development, it may be of any area of research. It has to take into account of the ecological, social, and economical impact. And if any research that we take into account, if it doesn't address these three areas, then it is going to be waste. And of course, it's a futile. That's what I always say that. It has to address these three areas. When I say, I mean, it is ecological, social, and economical. That's what really it's a need. When I say ecological, without forgetting the ecology, of course, the human race cannot exist in this world. We have to take into account of the sustaining the ecological factors. When I say ecological, it covers the entire species. It covers, I mean, it's a nature. In large, it's a nature. Nature has to be preserved. When you try to modify the nature, then all kinds of bad things happen. That is what we are experiencing. Either it may be, I mean, the corona that we are worried about today, or some few years back we have been talking of various other viruses, and today we are also talking of the natural disasters like volcanoes, or maybe the ices which are melting, or what do you say, tsunami kind of a thing, etc., etc. Why this is happening? That means, as a human race, we have failed to maintain the ecological sustainability. And naturally, when it is like a balloon, from one side, if you try to give a pressure, and the other side it will bulge. The same thing, that is what happening in reality today. Of course, when it comes to the social aspect, that's where we have to play a larger aspect, because it's our own living, our culture, the social balance that we have to maintain. Whether we have done it properly or not, I do not know. I mean, it's a question, is $1 million question, and we have to really think, particularly the researchers in this area have to think about it. Particularly when we talk about biosciences, that's where we talk of the social relevance and sustainability of the society, particularly our own living. And this has to be addressed. And only when these two are, ecology and then social aspect has been met, then the economy comes into picture. When these two are down, there is no question of talking of economy. Today we say that the economical situation of the entire gone back 20 years or 30 years. And of course, some stalwart of the World Bank has predicted, if this will continue, I mean, the corona will, if it continues for 2 more years, then we will go back by 30 years in the economical conditions. It's true. But who is responsible? It's only we. The human race is responsible. We try to compete and we wanted to win over everybody. With this competition, we try to invent many disasters. I think now it's a right time for us to think how to come out of these critical situations. It may be the biotechnological inventions by which our human race can be effectively saved. It can be a pharmaceutical, it can be your, antibiotic developments that you do, or how to improve your immunity system, or how to make, give a better health care. All that comes into picture. Of course, in another time, I think I was even talking about the research that the biotechnology has opened up new vistas of healthcare. And instead of developing our system and helping the human society to get benefited out of it, we are also doing some negative things. I think that has to be stopped, and it has to be a holistic, a 360-degree development, so that we can have the sustainability factor, both ecologically, socially, and which will lead to the economical sustainability. I am confident that this particular conference, particularly these specialists, the speakers who will address on these issues, so that the next generation of the society, that means the student community and the researchers, they will take a lead and will start working on this, so that we will rest assured we will not have difficulties of the corona period in the days to come. That is what it has to happen, and I'm sure the conference will address these issues, so that the society, the human society, will live without any kind of a fear. And of course, we can think of our ecological factors, social factors, and economical factors will all help us to grow further in our lifetime. So let me congratulate the organizers for having thought a really a fitting title for today's conference. I would like to really appreciate them, and also appreciate them for having organized, of course, with a good number of participants. And at the same time, I would like to appreciate the participation of the students and also the research scholars who have come forward to present their ideas and also listen to the experts. And it is with you, rest, and you have to do a lot of work. See, like in a short span of time, people have developed vaccines for the COVID. Similarly, many things have to be done in the future, and the whole world relies on your work and your contribution in the days to come. I'm sure that you will gain knowledge and then contribute to the betterment of this society. Let me wish you all the best, and thank you for the opportunity that you have given to me. Thank you so much. Wish you all the best.
Thank you, sir. A great person attracts great people. Here, I invite Dr. V. Kumaravel, sir, Director Academic of Vikas, to give the felicitation address.
A very good morning to all of you. My voice is audible?
Yes, sir. Super, yes, sir. Yes, sir.
Thank you. Good morning to our Executive Director and our Principal, sir, and all the resource persons, those who are going to deliver their lecturing in Sustainable Development School in Biosciences. So this is what a second international conference organized by our Department of Biosciences. So in this present scenario, the topic is most relevant today due to COVID and other problem. So we can able to discuss what will be our future, what is the role of bioscience professor and scientist and a researcher, how they are going to find solution, remedies to the present various problems due to corona and other natural issues. The Sustainable Development Goals, also known as the global goals, were adopted by all the United Nations member states in 2015 as a universal call to action to end poverty, zero hunger, and good health and well-being, and quality education, energy, economic growth, inequality, climate action, biodiversity. So, like these goals are very much important to have a peaceful living by the people all over the world. So in this present scenario, the role of biosciences and life sciences researchers is very much as needed in the present scenario. So this international conference is a very good platform to our students and the faculty members, so the researcher, to share recent research outputs and the future research tracks in India. So I appreciate the organizer to select a very good topic like sustainable goal. So all the students and the faculty members and the researchers, you have to make use of this conference in very well. So I wish you all the best, students and faculty members, to have a great success in this conference. Thanks for the opportunities given to me. Thank you.
Thank you, sir.
Thank
To
you.
be
Thank you.
inspired is to
To be inspired is great, but to be inspiration is an honor. Now, I would like to welcome Dr. T. Shankar Sir, Dean of Biosciences, with us for the program introduction.
Yeah, good morning to everybody. On behalf of Vivekananda College of Arts and Sciences for Women, Tiruchengood, and Vivekananda Arts and Science College for Women, Sangagiri, I welcome you all for the International Conference on Sustainable Development Goals in Bioscience. In 2030 Agenda, along with its 5 Ps, the 5 Ps, people, planet, prosperity, peace, and partnership, and 17 Sustainable Development Goals, namely no poverty, zero hunger, good health and well-being, quality education, gender equality, clean water and sanitation, affordable and clean energy, decent work and economic growth, industry innovation and infrastructure, reduce inequalities, sustainable cities and communities, responsible consumption and production, climate action, life below water, life on land, peace, justice, and strong institutions, partnerships for the goals. So likewise, 17 Sustainable Development Goals were formulated by the United Nations, sets out an aspirational vision for the future sustainability of our planet. Sustainable development involves diverse and complex approaches. Human stewardship of the planet, in particular its biosphere, is wanting. The trajectories of deterioration of critical features of the biosphere, loss of biodiversity, climate change, desertification, unbalanced nitrogen and phosphorus cycle, water quality and quantity, and the quality of the human condition, hunger, poverty, regional conflicts, refugees, human trafficking, rising health costs, diminishing urban security. On the other hand, and the institution of effective corrective or mitigating actions on the others are divergent, so planet Earth and human life are becoming increasingly unsustainable. Sustainable development in development that meets the need of the present without compromising the ability of future generations to meet their own needs. To counteract this divergence and to reorient evaluation of the biosphere towards a more formulated by the United Nations that takes into account the fact that all the key biosphere and relevant human behavioral processes are interconnected, interdependent, and hence must be steered via a systems approach. As a consequence, Sustainable Development Goals are exceptionally diverse, ranging from poverty elimination to moderation of climate change via safe cities, sustainable use of aquatic and terrestrial systems, to adaptation of renewables, as all the goals encompasses environmental, economic, and social aspects. Efforts to achieve sustainability are of necessary highly complex. Microbes are also biological stewards of planetary health and sustainability. Microbes are the predominant forms of life on the planet, both in numbers and total biomass. As the first forms of the life on Earth, they have evolved and exhibit a spectrum of evolutionary, functional, and metabolic diversity that vastly exceeds that of all the organisms in the tree of life. The ability of some microbes to inhabit hostile environment incompatible with most forms of life means that their habitats define the extent of the biosphere, and delineate the barrier between the biosphere and geosphere. And as microbes survive extreme environmental challenges, it is plausible that they allow life and hence the biosphere to recover after the major catastrophes suffered by the planet Earth that causes the mass extinctions. Example, deep sea microbes were probably large protective from devastating heat or cold waves in the atmosphere could survive for very long period in the absence of life. Microbes are very much our past and our future. Their ubiquity throughout the biosphere and the diversity of their activities make microbes a pivotal agents of planetary and ecosystem functioning. They mediate and regulate biogeochemical cycles and recycling of biological materials and waste, constitute key producers and sinks of greenhouse gases, and are thus important determinants of climate change, play essential role in soil structure and fertility and in the quality of protection of land, seas, lakes, and rivers. Microbes, therefore, are also a key members of committee to steward of planetary health and sustainability. Microbes provides a wide range of services to humans, animals, and plants. Microbes cover the surface of all other organisms and occupy internal and even intracellular niches in some, and influence diverse physiological activities of their hosts, including nutrition, health disease status, and health well-being. The microbial flora of an organism is designated its microbiome. The microbiomes of food animals and crop plants regulate productivity and thus the global food production and quality. Human microbiome, which has been termed as human services and thereby has a pervasive positive influence on our well-being as we discover when their ordinarily benign network activities become disrupted. Example, the antibiotic treatment. Our intestinal flora helps to digest our food and extract the nutritional content, and additionally provides us the essential nutrients. We neither make ourselves nor take in via our diet. It also plays a pivotal role in the development of our immune system and profoundly influence its functioning once developed. This is also the case in the animals and plants. On the negative side, a minority of microbes are pathogenic and able to cause disease. However, earlier notations that infections results through acquisition of microbial pathogens is now moderate by the knowledge that many microbial pathogens are part of our normal, ordinarily being flora, general only causing problems when our microbiomes become perturbated. The microbiome is the principal groups of microbes on the body surface and hence of pathogen colonization events that precede development of disease. Microbial pathogens colonization, population expansion, and subsequent interactions with the target tissues and immune system, which may or may not result in overt disease, are thus the classical ecological process, no more, no less. As a consequence, a system understanding the ecology of pathogens, particularly those with alternative lifestyles in the environment, is central to the effective interaction, management, and control. The current explosion in pathogen ecology research, example, environmental microbiology, reflect this. Recent findings suggest the Intestinal microbes may also profoundly influence brain function and are stimulating exceptionally exciting research at the interface of microbiology, neurobiology, and psychiatry. So this research promises to yield entirely new concepts in the perception of brain development and aging and the causes of management of mental ill health. So on. I think this international conference on sustainable development goals in bioscience will enrich your knowledge. Thank you. Thank you, Ananda.
Thank you, sir. An attitude of positive expectation is the mark of superior personality. Here, I would like to welcome our today's chief guest, Dr. Arun Mayuradan, sir, to give the inaugural address.
SGP sir? SGP sir? Is SGP sir available?
Yes, ma'am.
Yes, sir. So I requested SGP sir to give the vote of thanks for inaugural function.
Honorable and respected Chairman, Sir and Secretary, Professor Dr. M. Karyotype, sir, and the members of the management, and our Chief Executive, sir, overall not here, but however their blessings and motivations helps us to sustain our achievements. I like to thank the Executive Director, sir, Professor S. Kupusamy, and for his motivating speech. And I would like to thank Professor Dr. B.T. Suresh Kumar, sir, the Principal, Vivekananda College of Arts and Science for Women, Autonomous. And I would like to thank Academic Director of Vikas and Vias Arts and Science Colleges, Dr. Kumar Will, sir, and the Dean Sir T. Shankar, Dean Biosciences, for giving their speech during this wonderful event. So I feel very honored to offer the vote of thanks on this occasion of the inaugural session of International Conference Sustainable Development Goals in Biosciences, organized by the jointly by the Bioscience Departments of Vivekananda College of Arts and Science for Women and Vivekananda Arts and Science College for Women. And we are fortunate to have the wonderful speakers in this event from different parts of the world, like Dr. Arun Mayuradan, sir, from Spain, and Sino Vasne Germany from Malaysia, and Muthuswamy Ramakrishnan from China, Bhaskar Javier from China, and Saravana Kumar Murugesan from USA. So I also thank these speakers who is going to present their and share their knowledge with us and benefit the students of biosciences. And I'm very much impressed and to deliver my thanks for our Executive Director, sir, who said that scientific research have to address the ecological, social, and economic aspects. So thank you very much, sir, for establishing the social relevance of this conference with the current situations, and made our student participants from our college and other colleges to realize their responsibility and take a lead role in this critical situation, and to save the human race and the environment. So hopefully your words will come true after this conference. I also thank our academic director and Dr. Umervel sir, for motivating the young bioscience researchers to execute their goals of the United Nations in terms of sustainable development for the uplift of mankind. Thank you, sir. And I also profoundly thank our Dean of Biosciences, T. Shankar sir, who has given a very brief outlook over the 2030 agenda of UNO and explaining in detail about the 17 sustainable formulations by United Nations and narrating the diverse and complex approaches involved in this sustainable development goals and the role of the bioscience research to solve the global problems faced by the mankind environment. And he also explained very well the pathology and ecology research and the influence of microbiology in it. Hope these things will justify the need for the bioscience research to focus on the sustainable development in the near future. Thank you, sir. So at the end, I would like to quote that science and everyday life should not be separated, as said by Rosalind Franklin. And everything is theoretically impossible until it is being done, as said by Robert Elning. So we, the research of biosciences, have ultimate goal to solve the human problem, global problems, to solve this problem, that is sustainable development biosciences. So I express my hearty gratitude to all the speakers of this inaugural session who has joined today with us, and also thanks the different people who are associating with us directly and indirectly and made this inaugural session of international conference a successful one. I thank all these faculties, the management members, administrative bodies, faculties, directors, principals, and above all the student participants from our college and other colleges for joining this wonderful event. Thank you one and all. Thank you very much.
Thank you, sir. Now it's time to move on in technical session. There are as many atoms in a single molecule of your DNA as there are stars in a physical galaxy. We are each of us as a little universe. It's right to light up the technical session. Now I invite our head department of biochemistry, Dr. K. Purni ma'am, to introduce our guest.
Thank you, Pradeepa. As our guest required time for getting ready, we are now moving into the technical session. It's God's grace that we have successfully completed the inaugural function. So I now take this opportunity to present the points about our guests. Well, good morning once again, and it's my tremendous honor to introduce the guest for today's function. And indeed, it's privileged to have the eminent Dr. Arun Meyyadagan sir, the Chief and Director, Cytogenetics Department, Euroespace Biomedical Research Center, Institute of Medical Science and Genomic Medicine, Spain. Sir has a high profile, and actually it takes time to give his curriculum vitae, and in short and sweet, I introduce about the sir. So sir has completed his UG Biotechnology in Pondicherry University in 2007, and PG Animal Biotechnology in Bharatiya University in 2009, MPhil in Zoology with specialization in Human Molecular Genetics in Bharatiya University 2013. Sir also completed his PhD in 2013 in Genomics Medicine in Camilo Jose Cela University, Madrid, Spain in 2018. Then sir has also completed PG diploma courses in nanobiotechnology, cheminformatics in Bharatiya University, and PG diploma in classical and molecular cytogenetics in European Cytogenetics Associations. Sir has sound knowledge in various techniques like chromosome culture, then karyotyping, then immunofluorescence, then our electrophoresis. Sir also positioned in various fellowships like the JRF. He has gained 2 JRFs. He has worked in identification of the cancer cells. Sir has one DST research fellowships, and also Sir has worked in neurogenetics as a research fellow. Currently, he is positioning the chief and director position in the Institute of Medical Science and Genomics Research. So Sir has more than 25 research publications in various journals with impact factor of more than 70. Then Sir has resourced in various organizations in international conferences, and Sir is the resource person in European Society of Human Genetics in clinical medical genetics, and he has been awarded with the Travel Fellowships Award in 2015 in Hyatt Regency Miami, Travel Fellowship Award in 2014 in Hyatt Regency Miami, and also Sir has international travel fellowships and summer fellowships also he has been awarded. He has visited more than 50 countries all over the world for various scientific events. Then he has 10 years of research experience as in most of the research events in related to cytogenetics, and he has headed more than 10 conferences and workshops. May we have the Dr. Arun Mayalagan sir for this presentations. Over to sir. Sir, Dr. Arun sir.
Yes, ma'am. Could you hear me, ma'am?
Yeah, you are available.
Yes. Thank you, ma'am. Thanks a lot for the introduction, and also thanks for the opportunity. I thank the management and also like your chairman, Professor Dr. M. Karnanidhi sir, for providing all the facilities conducting this international seminar through virtual mode. And it's been a great pleasure during this COVID-19. You could able to conduct an international conference that will be benefit for the students and for other faculty members, and it's been a great pleasure. And I'm honored to give a session, and it's been one second, ma'am. I have some technical issues over here, and I'm sorting it out. I'm you can able to view the screen, no, ma'am? Now?
Yes.
Fine. Okay. So thanks for the opportunity. I would like to thank Purni ma'am for giving me this wonderful opportunity. And she has been following me for 2 weeks, and it shows how much interest she was having towards the students and also about the conference. And it's been great pleasure, and thanks a lot, ma'am. And this is my overview of my institute, and where you could see the right side is the hospital and left side is the research center, where there is an this is the overview 180-degree view of our institute. So today we are going to see about human molecular genetics. It will be a general introduction and update, because I came to know that many undergraduate students and postgraduate students are attending this conference. So I hope that if I speak about many basic things about cytogenetics, I hope many future budding scientists who are attending the session could able to jump into the genetics field. So that's my motive for this talk. So see, this is my first slide. Wherever I present or wherever I go and give some invited talk, this will be my first slide, because this slide has a connection of some 10 years back, 10 years back, because like this was the slide that I could see in the Google search. So like I have a mentality to know about the DNA structure and the combination and everything. So just I'm eagerly when I was doing my post graduation, I want to know what's inside the nucleus, how it works, and how far it plays a major role in the human body and other things. So that tendency make me to select my core area, cytogenetics. So this was the slide which you could see, like I'll climb up this strand of DNA to see where life takes me, and the life takes me here as a scientist, of a cytogenetics scientist over here. So generally, you students should have the tendency to reveal the secret behind everything. That is the core idea of a scientist. I'm not saying about genetics alone. If you are interested in environmental monitoring or cell biology, anything, you should have the tendency to know more about the basics. So basics is more important than anything. So the basics plays a major role. So those who are not up to the mark till now, like, you no need to be worried. You should be more strong enough in basics. When your basics are strong, then your future will be more bright. So I'd like to give an explanation about cytogenetics. Cytogenetics is the combination of genetics and cytology. You know what is the definition of genetics? This is a branch of science which deals with the study of heredity, and the variation is called as genetics. And cytology is the branch of science which deals with the study of structure and function of cells, and cell organelles is called as cytology. Here we are going to study about the chromosome structure and other things. So that is so-called cytogenetics, the branch of science, the branch of genetics, which deals with the study of cell and structure and function of the chromosome is called as cytogenetics. So these are some small details about that. The study of chromosomes and the related disease states caused by abnormal chromosome number and or structure. And chromosome is nothing but it is a complex structure located in the cell nucleus, composed of DNA, histone and non-histone proteins, RNA, and polysaccharides. And cytogenetics involves the study of chromosomes, as I said earlier, and both the number and structure of the chromosomes are analyzed using the cytogenetic techniques. So whenever we whenever we get into some topic, like we want to know the origin and history of the chromosomes and other things, how it's evolved and how the cytogenetics patterns becomes more popular, and where they have nomenclatured all the things. Nomenclatured in the sense, like, there are some scientific terms that we should use in this scientific community. Think like we can't say, like, we have our own language. We can say, like, for example, you will be studying about this botany family, you know, like the Fabaceae, Asteraceae, and other things and all, because like when we say in common term, these things will not be understood in other parts of the country. So when we are speaking about, like, the scientific term, then everyone can understand that. So to understand everything in common or in scientific writing, these things should be nomenclatured. It should be they should have a group, and the science group will be there, and there all the nomenclature will be taken part. So we'll see the origin and history, how it evolves and how these things happens. So generally, the chroma means color, and soma means particle, which means it is a color particle. And it was the first description given by a scientist, Carl Wilhelm von Nageli. He found the plant chromosomes, and he named it as chromosomes for that particular color particle. And it was the description that takes place. It was the description, or this chromosome term comes in the year 1842. And generally, like, we now know that we have 23 pairs of chromosomes, that is 46 chromosomes, that is 22 pairs of autosomes, and 1 pair of allosomes, that is body chromosomes, and 6 chromosomes. 22 pairs of body chromosomes, 1 pair of 6 chromosomes. 6 chromosomes is X, Y, which differentiate male and females. The sex chromosomes differentiates male and females. So earlier there were many confusions in scientist community, that the males has 47 and females have 48 chromosomes, and whether, and some says 46 chromosomes and some says 48, and some scientists say 47. And these are the theories that goes on in chronological wise. Like, see, in the year 1921 they say it is 46 or 48. We never know that. In the year 1923, the scientist so-called Winnie Warter, he said like 48. The human has 48 chromosomes, and then comes because the reason behind that is when we see the chromosomes in the microscope, it is in a clumped nature. Before 1956, before the invention of the exact number of chromosomes, and it was an accidental invention to find the exact number of chromosomes, because they were doing this. This scientist John Levin, everyone know the father of genetics is Mendel. No one knows about the father of cytogenetics. The father of cytogenetics is Tijo and Levin. So they were the one who found the exact number of chromosomes. So it was an accidental invention. They were doing experiments for this identification of chromosomes, and during that time they have an KCl solution. We say the KCl solution is an hypotonic solution, because when the KCl goes inside the cell, the osmosis takes place, and the cells will be in a bulged way, like a balloon. It will change like a balloon, because the osmosis reaction takes place when the KCl enters inside the cell. So when they are doing experiments, they were having some KCl solution for some other experiment, and during that time the KCl solution has been added accidentally. They never know that the KCl solution has been added, but it has been added accidentally, and then they could able to clearly differentiate the chromosomes. And this is the first picture of the chromosomes that has been taken, the 23 pairs of chromosomes, which you could able to see very clearly over here. And this was the structure, and this is Tijo, and this is Levin. So this is the first picture, which you can count the full complement of 46 human chromosomes. So then comes identification of the genetic disorders and other things, because this is so-called a new medical field, the human cytogenetics, in the year 1959. Because they confirmed that the chromosome number is 23 pairs, that is 46 chromosomes. And suddenly, a scientist called John Langdon Hayden Down, when he was doing some chromosome classification with a patient, with a Down syndrome patient. That time he never knows the patient name. The patient has Down syndrome. Okay, so it's so-called, earlier they so-called, the common term is mental retardation. Nowadays we should not say them as mental retardation. We should say them as a special person or special child. We should name them like that. This is for understanding, I'm saying it. So when you see someone who is mentally retarded, we should call them as a special child or a special person, like that. We should not call them as mentally retarded. One or the other way, we all were mentally retarded, so we should not call them as mentally retarded person. Okay, so during that time, he was observing a boy who's that time is mentally retarded. So they were doing classification for that boy, and where he could able to not accidentally, where he could able to find 47 chromosomes. And everyone was shocked, they were saying 46 chromosomes, how it is 47? And then they come to conclusion, it is so-called Mongolism or Down syndrome. The Down syndrome is so-called by the scientist name, where you could able to see here. So the term Down syndrome came from the scientist name, and he invented the classification and other things for this Down syndrome. Not classification, the extra chromosome in 21, in 21st chromosome. So there were many common terms for this. We say as 21XXY chromosome. So there were many common terms for that. So this was identified by John Langdon Hayden Down. And this was the article that's been published by him, that this Down syndrome patients will be having 47 chromosomes. That's what like the genetic disorders and other disorders will be identified using this karyogram. Nowadays, we have many genetic markers, but some five years back, we have to take a amniotic fluid from the pregnant woman, and we have to find it out whether the baby is having this Down syndrome or not, if they have any suspicion through this genetic counseling. So again, this was the article about the other disorder, Edwards syndrome. So 1950s, we used to say the golden age of cytogenetics, where we could able to find Klinefelter syndrome and Turner syndrome, the loss of chromosome, and trisomy 13. There will be a 3 chromosome in 13. It is the another name for trisomy 13 is Patu syndrome, and trisomy 18, and another name of trisomy 18 is Edwards syndrome. In chromosome 18, we can able to find 3 chromosomes. We will see the nomenclature in the future slides just to know that. And Philadelphia chromosomes in CML. CML is it's a chronic myeloid leukemia. The short form I mentioned is CML. The CML can be identified only by cytogenetics, not through any modern techniques. We can't able to find any there is no any modern techniques like NGS, anything can't be identified, because the translocation can be identified only using a cytogeneticist. So that is a need of a cytogeneticist. No, the molecular biologist or the genetist can't able to find the translocation, because now we have many modern systems like NGS and other things. We put it and we can able to see the deletion, insertion, anything. But the translocation alone, like we have to depend on the cytogeneticist to know it, the translocation takes place or not. So as I was saying, like there is a nomenclature. There are some nomenclature. In the year 1960, Denver is a name. Denver is a country name where a group of scientists has been gathered there, and they were creating some names, the common technical terms, terminology terms for this chromosomes, or the arrangement of chromosomes, or all these things. So first, in the year 1960, in that Denver conference, they were saying like there were sorting criteria for human chromosomes, like biggest first and metacentric first. I'll explain what is mean by metacentric, submetacentric, acentric, everything in the future slides. And in the Chicago conference in the year 1966, they named like a short arm is P and long arm is Q. And I'll explain why it is named as P and Q. P is petite. Petite is small in French. And Q, like it is so-called small and big. So they named it very easily like P and Q. P has a meaning, Q has no any acronyms that. So this was the first chromosome, this was the first nomenclature. This is a pictorial presentation. We use like ISEN, International Systemic Committee for Nomenclature, for this particular chromosomes, and they have from 1 to 22 they nomenclature like this. So this is how it looks, the nomenclature of the chromosomes, the patterns. And this is a pictorial presentation. It's not an exact chromosome. The chromosome never looks like this. This is the pictorial representation to understand very clearly. So coming to the karyotype. Karyotype is nothing but chromosomes are usually all jumbled up and mixed together in the nucleus of cells. This is when we see in the slide, it will be like this. But as a cytogenetist, we have to arrange the chromosome like this. This needs more experience to identify or to nomenclature that. It will be arranged according to their size and shape. The chromosomes should be arranged according to their size and shape, which is so-called karyotype. It has been arranged like this, biggest and metacentric, submetacentric, like that. But we'll be seeing in the future slides how the metacentric, submetacentric chromosomes looks like. But I'm very sure like you are aware of all these things. The short arm of the chromosome, that is C. The short arm of the chromosome is called as P, and the long arm of chromosome is called as Q. And the bands are numbered independently on the short and long arms. I'll explain the band and other things and all. See, this is how the position of centromere takes place in the chromosome. See, this is centromere, and if the centromere is in the middle, P and Q has same distance. It is so called metacentric. The chromosome 1 is metacentric, and chromosome 2 is submetacentric, where you could see Q is bigger, and P is little larger. It is so called submetacentric. So 2 to 5 chromosome will be submetacentric, and the fifth chromosome is very submetacentric, where you could see a Q region is a little bigger and P is little smaller. So this is so called very submetacentric, and the acrocentric chromosome is Y chromosome and 21st chromosomes, okay, and X chromosomes, not X chromosomes. And acrocentric, where you could see the centromere alone, there will not be any P arm. The P arm will be very, very, very small. That is so called acrocentric over here. This is the position of the centromere. If you could able to understand the position of the centromere, at least we could able to arrange the chromosomes like this, like 1 to 22. But we should have a clear understanding. And this is the classification of the chromosomes, like chromosome A, chromosome 1, 2, 3, which will be like largest metacentric, where you could be able to see this metacentric over here. So this is so called group A, and group B is 4, 5. Generally, when we see, like, we could see a mutation in the group B, that means like chromosome 4, 5, maybe 4 or 5, there will be a deletion or something. We'll be saying that abnormalities. Okay, chromosome 4, 5 is like large metacentric, and chromosome 6 to 12 is submetacentric, that is group C, and group D is medium acrocentric, that is chromosome 13 to 15, and chromosome 16 to 18 is like submetacentric again, it is group E, and chromosome 19 and 12 is short metacentric. There will be like an acrocentric kind of thing, but it will not be exactly looks like acrocentric. See, the acrocentric looks like this, but the short metacentric is like this. And group G is chromosome 21 and 22. That is it will be very short acrocentric. See, these are the chromosome terminology. We have seen about A to G is the chromosome groups, and 1 to 22 is the autosome numbers, and XY is allosomes. Allosome is nothing but the sex chromosomes. For deletion, we will be using a short term as DEL, and for duplication, DUP, and inversion is INV, and short arm of the chromosome is P, and the long arm of chromosome is Q, and the satellite chromosome is S. These satellite chromosomes can be able to identify when the person is occupationally exposed to any radiations or anything like that. If you take a blood sample of a person who is working in any kind of radiation, like in the nuclear plant workers or the nuclear reactors, or exposed to any kind of radiation, we can able to identify a satellite chromosome. If we identify a satellite chromosome in any of like any person, then we can able to say that they are occupationally exposed to any kind of radiations. And the translocation common, like the terminology is T, and sometimes we use plus minus before the chromosome number. It indicates addition and deletion. Minus is deletion of the whole chromosomes. This is the chromosome terminology. And this is the inheritance of gender. We know like how the male and like the boy and girl is formed due to this fertilization. Like the mother sex cell is XX and the father is XY, the genders. And you know about the meiosis reaction, how it takes place, because I don't want to explain more about meiosis, because you were studying from 10th, like almost from 8th, about the meiosis and mitosis things. To understand more about this thing, like you should be strong enough in understanding the meiosis and mitosis. So this is how this thing, like you also know about the Punnett square, no? Like this green piece, short, tall, the mechanism behind this is same, XY, XX, and by that we can able to say like the chance of a girl is 50% and the chance of a boy is 50%. This is the sex cells. You have this thing in the mind. The female has XX and the male has XY, and there you will be seeing many kind of questions. The many kind of what to say the questions in the exams like this. How will you differentiate male and female, XX, XY, like that? And even we have 6 chromosomes of animals because we have animal genetics, plant genetics, and we have many kind of genetics that's been going on. And for each animals, we have a different kinds of chromosome nomenclature too. And see, like this we no need. And for the birds, amphibians, reptiles, the butterflies and moths, we used to say, like, for the male it is ZZ and female it is ZW, and for the grasshoppers it is XX and XO. Just to know, like you could know what are all the sex differentiation in different animals. In some articles or in some common explanation, you could see like they will mention as ZZ or ZW. At least by this time you could able to know they are saying about male birds or male amphibians or male butterflies, like that. So now we comes to the part, like the most important part, how we extract chromosome from the body. So where the chromosome is present, you know that there is a blood cells. Inside the blood cells we have endoplasmic reticulum, Golgi complex, everything, and inside the nucleus we have the DNA. Like DNA is like it is a tightly coiled structure, and inside that we have chromosomes, and we have to isolate chromosomes. But how we will be isolating the chromosome? This technique is identified, not identified. We follow this technique, like Moored et al., Moored et and Hungerford. This is the standard protocol from the year 1960, where we find an well-spread chromosomes plates for detailed examination of metaphase. You'll be confused by what is meant by metaphase. So you should be strong enough in understanding the mitosis. And I don't want to explain more about mitosis. You know about mitosis also, because if I take mitosis, then the whole 1 hour will be over. Mitosis, like just I give a brief explanation about mitosis. You know that there are telophase and the other phases that's been in the cells. So in the metaphase only, during this metaphase stage only, we could able to identify the chromosomes. So what we have to do is, like, we have to grow the cells inside the culture tube, like inside the culture vial, for 3 days. For getting this metaphase stage, it like the metaphase that happens in this happens in the cell is like 48 to 72 hours. So what we have to do is, like, we have to grow the cells in a culture tube. What are all the requirements that has been given in our body to maintain the cells in our body? We have to give all the requirements in the culture tube to make it grow, so that we can able to induce the mitosis in that. And then when we induce mitosis at the after 2 days, like after 48 hours, where the metaphase stages starts. So when the metaphase stage starts, what we have to do is, like, we have to arrest the mitosis reaction that's been going inside the tube. And then we have to start the experiment, and we will be doing all sorts of experiment to view the chromosomes. So this is the principle behind that. So these are all the chemicals that we need. This is so-called RPMI. RPMI is nothing but Roswell Park Memorial Institute. This institute has a this has a medium. I will show how it looks like. The RPMI looks like. And fetal bovine serum is like a growth factor. This is taken from the calf serum, where the growth factor is more. So RPMA has a more number of constituents, which helps in growing the blood cells inside the culture tube. And the phytoheme agglutinin, which is a mitosis inducer. If you had phytoheme agglutinin, like the mitosis growth factor takes place. And we will be adding antibiotic and vitamins to avoid the contamination in the cell culture, because it is more what to say, like we should be more cautious in doing the experiment. The contamination should not take place. If the contamination takes place, the whole experiment goes wrong. And doing this experiment is quite tedious. Like it takes 3 days. So always the culture lab should be clean, and when we are doing the experiment, and everything should be sterilized, everything should be in a proper way. If any contamination takes place, the whole experiment goes off, and it is a 3 days experiment. If something goes wrong, then your hard work will be lost, and the chemicals are very costly. So to maintain that, to make it in a proper way, the cytogenetics lab should be in a sterilized way and should be fumigated regularly. And as I was saying, like 0.075 m KCl, because the hypotonic treatment, this plays a major role in identification or the numbering of chromosomes. If the KCl treatment is not proper, then your experiment goes completely wrong, because you can able to see the cell, you can able to see the chromosomes, but you can't able to differentiate the 23 pairs of chromosomes. And colchicine is like a mitosis inhibitor. As I said, like we have to stop the in the mitosis reaction, we have to stop this stop in the metaphase stage. So to stop that in the metaphase stage, we will be adding 0.01% of colchicine, and mainly we need a blood of 0.5 ml of blood or 30 drops of blood, and Jimsa stain to view the chromosomes, because like when you see the chromosome in the slide, it is colorless. So to view that, we will be adding Jimsa and trypsin. And absolute methanol and glacial acetic acid, it is so-called fixative. To fix the chromosomes in the slide, this fixative is important. So this is how this is the short mechanism behind that. The cells are stimulated to divide and are incubated for 48 to 96 hours. Cells are arrested at mitosis. Cells are dropped onto glass slide, and using G banding technique, we can able to identify the chromosomes. See, this is the preparation of metaphase, where you could able to see the laminar airflow. We used to work inside the laminar airflow. This is a closed room, and inside the closed room, we have the laminar airflow. I hope like you could see the laminar airflow in the microbiology lab, because to avoid the contamination, we used to work with that. There will be a HEPA filter and everything will be over there to know whether it is the laminar airflow is working exactly or not. And I'm not getting into this laminar airflow mechanism, just to know why we are doing inside the laminar airflow. And where you could see this, this is so-called antibiotics. This is RPMI, and this is vitamin, and this is FBS, and we'll be mixing all this in this culture vial, and we'll be adding 0.5 ml of blood, and we'll be incubated, and we'll be incubating this culture vial in the incubator at 37 degrees C. That is 36.8 degrees C, because the body temperature, the body temperature of human body is 36.8 degrees C. So we'll be maintaining this 36.8 degrees C, which is connected with the CO2 incubator, which is connected with CO2, the so-called CO2 incubator, and we'll be keeping here. And then we will be tracking this culture for 48 hours, and after 48 hours, we'll be adding colchicine by 62nd hours. So where the metaphase will be clearly seen, and it depends. Someone will be adding at 68 hours, someone will be adding at 72nd hour. So it varies to the according to the lab standardization. So I used to add between 62 to 66th hour. I'll be adding, and I'll be incubating for 1 hour, because the reaction should take place to stop that at the metaphase stage. And then I'll be doing this with the harvesting process, we used to say, by adding the KCl. Like we'll be transferring this solution into this falcon tube, you could see here, and then we'll be centrifuging it, and we'll be removing the supernatant and the pellet, where you could able to see the blood cells alone, and we'll be adding the KCl solution. And then after that, the same procedure will be following it up, and then we'll be adding fixative. And after adding fixative, we'll be preparing the slides. The slides will be like this, and and we'll be staining the slides. And when we stain the slides, we can see there are different kinds of staining. I will say that also, because see, I can't explain in detail about this experiment because it takes almost an hour to finish the experiment. At least you'll know a brief idea how to prepare this chromosome alone. And these are the different kinds of staining that we are following: Q, G, R, and C. G banding and R banding is the banding that we use to follow all over the world, and the R banding is preferably following in France, and the GT banding is followed in all over the world, in India and in US, in Europe, everywhere. But in some labs, we follow the R banding. R banding and G, there is no difference between G and R banding. The reverse of R banding is G banding, and the reverse of R banding is G banding. Okay, so it is the reverse process. But see, like for G banding, we will be using GINSA. For R banding, we will be using there will be a kind of technique, like not technique, with the solution will be totally different. And the C banding, where we can able to find the centromere positions very clearly, and the Q banding is like we can able to see the bands alone. I will show in the next slide. See the Q banding, like where you could see the bands, and the C banding, you could see the centromere position alone very clearly, where you could not see any banding things. And here you could able to see the clear image of this banding. You could see banding. Here the resolution is not proper, but I will show it here. This is the G banding. The reverse of G banding is R banding. Okay, there is no any confusions. Here the dark region will be the light region in the R banding, and the light region will be instead of light region, there will be dark region in the R banding. That is the difference. So this is the G banding. I follow the GT banding. Here we say, like, see, this is centromere, this is a heterochromatin band, and this is telomere, and this is euchromatin. The white region is euchromatin, and the dark region is heterochromatin, and the top position and the lower tip position is telomere, and this is the centromere. This is the morphology of this chromosome that we can able to identify the deletion, duplication, and anything. This is how the chromosome looks in the light microscope in 100x. So this is the normal karyotype. See, we get this, and then we will be differentiating using this ideogram. We have a software for this. Earlier, there were no softwares during my graduation or during my pre-doctoral degree, that is MPhil. We don't have software in our lab. So just we will be seeing, we will be taking a picture and we will be arranging ourselves using these kind of techniques, ideogram. So this is by software where the picture will be taken, and the picture will be taken to that software. And using the software, it will differentiate. This is first chromosome, second chromosomes, and it will automatically segregate the chromosomes. And the main thing is that the slide should be very clear, so that only like we could able to the software can able to perform very clearly, or else we ourselves has to pick and we have to keep it there. This is a typical normal male karyotype CXY and female karyotype XX. And this is how it looks like this traditional in the traditional microscopy. This is in 40x, it is so-called metaphase. And when you from 40x, if you zoom it into 100x, this exactly it looks like this in high power. See, this is in GIMSA banding. This we can able to see in the in the software. And when you add GIMSA, when you see in the light microscope, the chromosome looks exactly like this. This is X chromosome and this is Y chromosome. This is the normal control male. And as I was saying, the satellite chromosomes, no, in the earlier slide, when any person is affected with any radiation, there will be having a satellite chromosomes. Even the mobile users and the houses, like there are many towers near our home, this Airtel network and other networks. And if this radiation is high, then there are many chances of getting satellite chromosomes in our body. So we should be very clear about that. That's what we used to say, that like there should be, the frequency should be less in the less in any kind of tower, because it has many potential carcinogenic effects. So this is how it looks, low and high resolution karyotypes. See, like this is so-called band, this is so-called region, and this is so-called band, and this is called subband. If you zoom, this is a typical example of 7th chromosome, and this is Q, it is long arm, so-called 7Q, and this region is called 2,1. And when you zoom the 7Q21, it is 7Q21.1. There is a band inside this. And when you zoom inside the band, you could see the subband, 7Q21.11. This helps in identifying the exact mutation part. And that is the main reason for explaining the slide or understanding this slide. This is how it looks, the chromosomes morphology with the software. When we give the patient's report using the software, sometimes we use these kind of thing to understand it, understand very clearly. So the morphology will be like this. This is the pictorial presentation and the morphology, and this is the chromosome numbering. By this, we can able to identify the mutation or like any kind of deletion or duplication in this. So the normal karyotype formula, like 46XY is male and 46XX is female. 46XX plus 21 is Down syndrome. This is how the report look like. Plus 21 is like we have extra chromosome on plus 21. That is 47 XXX or 47XX plus 21. In 21st chromosome, we have 3 X chromosomes. And 45XY is like Turner syndrome. And 46XY 5P deletion. And 46XX3 translocation, this is like CML, like for the leukemia, and 46XY deletion 15Q2, like this is like a typical example for any cancer samples. So this comes under like we have to know recessive inheritance and dominant inheritance. These two things plays a major role in understanding the genotype and phenotype correlation. So if a father, if you have a carrier father and carrier mother, think like in their generation they have retinoblastoma or any kind of diseases, and there are chances of getting 50% of carrier and 25% of normal and 25% of affected. This is recessive inheritance. The dominant inheritance is like an affected father and a normal mother. That is like if see like a father has a cancer and the mother is very normal, and think like there are chances of 50% of normal and 50% of affected offsprings. In this case, we can say that if it is normal, there are no chance of the passing of offsprings. There are no chance of carrier thing. But in this case, like there will be a carrier. That's what we used to say. Like generally, we will say it is a genetic disorder, and we'll say like this has been passed from your offspring. So to understand the genetic counseling and other things, we have to be have a clear vision about the dominant inheritance and recessive inheritance. So if you are clear enough in that, then you can able to identify the genotype and phenotype correlation in the family tree. Generally, when we are doing some karyotype or any kind of genetic studies or the cytogenetic studies, we will be putting these kind of genetic or the genetic morphology. So why cytogenetics? Like these are all the basics, but why cytogenetics? How the cytogenetics plays a major role. In cytogenetics, like cytogenetics helps in identifying the locus, because see, we straight away if we go for finding the gene, no, it takes an enormous process. We can't able to identify in which chromosomes the gene has been mutated. If suppose think it is a new disease, okay, in new disease we can't suppose think new cancer. In the new cancer we can't able to identify the gene. We can able to identify the gene, but it is a cost effective process. To reduce the cost effective process, you have to identify the locus. If you identify the locus, then the cost effective process will become more or less. These things are like for your age, like it will be little complicated. At least try to understand what I'm saying. Like when you are studying or anything, it will be quite helpful. Like in near future, it will be helpful. So for identifying the locus, the cytogenetics plays the role. When we're identifying the locus, like we can able to find the mutated gene. If you're able to find the deletion part in this, you can able to identify the gene part over there. And then if you're able to find the mutated gene, we can able to understand the pathobiology behind that. And then we can identify the target. And then we can go for the treatment. This work is by a cytogeneticist. This work is by by a molecular biologist or a genomic person, or this one is by biochemistry, and this one is by a bioinformatician or any kind of genomics and proteomics kind of work who is doing. And the treatment is finally to a medical biotechnology. See, this is the combination of all these things. But most of all, like the cytogeneticist plays a major role to identify this. So it helps in identifying the locus. If you could able to identify the locus position, the deletion or the duplication position, this will help in finding the mutated genes in that particular part, the molecular biologist. So it is a kind of a teamwork. So that's why the cytogenetics are priorly important. Okay. So this is how this process works. It's about the brief explanation how it works, how this system works. Okay. So this is like an common information, like this is to improve the detection or anything. There will be an advancement in the techniques, no? Like with the help of the GT banding, we can able to find only the large kind of deletion, but we can't able to identify the smaller deletions. So for the smaller deletions, we have different kind of techniques called FISH technique or the CGH technique. And these are the techniques that helps, and microarray technique. These are the techniques which helps in identifying the smaller deletion in the chromosomes. So that's this slide explains that. And see, like this is like fishes, like fluorescence in situ hybridization. I will show that. This helps in that, like for aneuploidy, like only we can, and by using karyotype we can able to find 100%. For the large deletions and the large duplications, translocations of large segments, the karyotype plays a major role. And the telomere fish helps in identifying this also, but the disease specific fish is not that. Okay, this cryptic rearrangement of telomeres, by using telomere fish we can able to identify it, not by the karyotype. And 1p36 deletion which could be identified using this GT technique with more number of experience and with a good microscope. And same like this, these are all the genetic disorders where you could able to see Prader-Willi, Williams-Buren, Karyotype, Angelman, and Smith-Magenis, and DeGeorge1. These are all the conditions, the genetic conditions that can be helpful helpful by using the karyotype. We can able to identify it, and some by the fish technique and some by the telomere fish technique. And this is so-called fish technique. Fish technique is nothing but a laboratory technique for detecting and locating a specific DNA sequence on a chromosome. If there is any mutation in a specific part of the chromosome, there we can use this kind of technique. It is a kind of a probe. I hope like many were aware of this PCR and other things where we'll be adding the probe, and it will be attaching to this reverse and forward primers like that. So same kind of probe will be designed for this particular thing. And if that particular probe gets attached, then we can able to say that they have a disease, because that probe will be designed according to this disease specific probe. If you are having a doubt, like there is a 13p marker, the 13p marker is for this breast cancer in women. So we will be designing such marker, and when we do the fish technique, we can able to identify it. And also, like as we are saying, like see, like for this kind of technique, like a 1p36 deletion or Karyotype. So for finding that also, like we will be having a probe. When this probe gets attached, like there will be a signaling, because all were fluorescently probed. And with the help of that, not fluorescently tagged, with the help of that, we can able to identify that whether they have a chance of getting this genetic disorders or not. So this is how the technique goes. And till that, what to say, the same technique that has been followed in doing this karyotype, no, the same techniques will be taken, will be done. Only like there will be a different steps that's been different after this fixation. So then like we'll be denaturating it, and then we'll be hybridizing. Once the cells has been placed in the slides, then with the slide we will be denaturing, and then we'll be going for the hybridization process, and the post hybridization washing and detection, and the counter stain, and then the visualization. This is the common procedure for understanding the fish technique. So this is the picture where it clearly explains how the fish techniques, how we used to do the fish technique. Okay, so again, I'm not going in detail because it takes time. Just to know an overview idea of this, I'm explaining what is meant by fish technique. And where you could see a very colorful detection. This we can this is helpful in explaining the different types of chromosomes. You see the first chromosomes looks like yellow color, and this needs an enormous work, and we used to say this is chromosome painting. So this is like a spectral karyotyping. Sky is helpful in identifying if a single patient has 2 or 3 mutation. In that case, this spectral karyotyping plays a major role. Same like fish technique, this technique also will be doing the same procedure, same as fish. Only the application of dye varies. Here we'll be adding more dye to find to differentiate different colors. This is how the spectral karyotyping looks like, but no one follows the spectral karyotyping because it is a very costly process and it is time consuming. So this is array-based comparative genomic hybridization. This process is same like fish, and then for the CGH, there will be a detecting image analyzer will be there. We nowadays like see like in our lab we used to get cystic fibrosis and dementia patients are more common over here. So we have a marker for this that will be having an that will be quoted in the array. So then we'll be doing the steps, and we'll be seeing a signaling like this. See, it is normal, and when we see there will be a duplication signal over here like above the line, and below the line it is so called deletion. This is the image of this CGA. By using the CGH software we can able to identify that. This is the standard and this is the patient one. Like you could able to see the standard, like it is plain, there we have no any signaling. If it is comes like this, it is plain, and if it goes like this, it is like deletion, and it is above there is signaling, it is duplication. It looks exactly like this. By this we can able to identify the deletion and duplication in the sample. So this is a typical examination. What are the differences between karyotype and the fish image, microarray, and the DNA sequencing? DNA sequencing, as I said, like it helps in identifying all the sequence changes and the single gene mutation except the translocation. And see, the karyotype helps in detects translocation. The translocation can be identified only by karyotype, not by any other modern technique. See, fish array, no, microarray, no, the DNA sequencing, no. No other modern techniques are involved in identifying the translocation. For identifying the translocation, you have to completely depend on a cytogeneticist. And for identifying the large deletion or duplication, like this 3 plays the major role. And to detect deletions or duplication in part of a chromosome, like the fish and microarray plays the major role. So these are all the differences between the karyotype and the fish image, microarray, and the DNA sequence. And now the present work that what I'm doing is like a pharmacogenetic work. There is a combination of a pharmacology gene and the genetic gene, and you just understand what is meant by pharmacogenetics. Because see, when we are taking some drugs, this drug will not be efficient for everyone. We can't say like the drug will work for everyone. Suppose think like I'm affected with some cancer. If they are giving a common cancer drug to me, it may respond or it may not respond. We never know that. So what we have to do is like every human has a different combination towards the pharmacology drugs. So we have a pharmacology genes. What we have to do in India, they are not performing like this. In US, in Europe, in many countries, they are performing this. But this is a very costly technique. When we are taking any kind of drug, there may be any side effects. So what we will be doing is like we have to give the drug and we have to find that whether these drug is responding to me or not. To me, not to the patients or not. So if the drug is responding, then we can able to identify using this pharmacogenetics panel. If the drug is not performing, then we should not suggest that drug for them. Suppose like when we go to any oncologist, like they will give you any kind of steroids and drugs. Before giving the steroids and drugs, we have to know that drugs and steroids are responding to us or not. Because without knowing that, we can't take the medicines. Because some will give solution, some cannot give solution. So for that, we have to know that the drug is responding to our body or not. That is more prior. Without knowing it, if we are simply taking the medicine, the adverse effects will be more. There will be damaging of kidney or liver, because every medicine has its own side effects. So that's what we are saying, like we have to reduce the medicines accordingly. So I am allergic to some drugs because I know my pharmacogenetic panel, how it works. I have some allergic to some kinds of drugs and I know it. So to identify that, we have to do a pharmacogenetic study, but it's a very cost-effective study. I don't think so. Like in India, no one is performing this kind of study. This kind of pharmacogenetics has been first explained in Mayo Clinic, which is present in US. And then later we have adapted this technique and we are doing that kind of study over here for this dementia or any neuro patients. Because for neuro patients, like we'll be giving many kind of epileptic drugs or all kind of drugs. So before giving the drugs, we have to know that whether these drugs are responding to them or not. By that, we can able to identify it. So these are some kinds of programs that's been useful for undergraduate students and postgraduate students. You can go for this summer training program during your summer vacations. And undergraduate students and post-graduation students, you can make use of this link. I will send this link to Purni madam so that she can circulate this. And just you should have a knowledge about that. When we are doing undergraduation, we don't have any knowledge about this. At least by this invited talk, like you should have a knowledge about these kind of websites that's been in India. Like they are giving some prestigious fellowship programs. This fellowship programs helps in knowing how the lab looks like, how the lab works, how the structure looks, how the research looks like. At least you'll know the basics before going to do your doctoral degree. So this research program helps in plays a major role. And also they give stipend for you, like it is a free program. All these programs, whatever I mentioned here, it is a free program. But the thing is, like you have to apply the application and you have to get a recommendation letter from your principal or your teacher, like the professor over there, and then you have to submit a proposal for that. So you can seek help from your professor and we can start doing your application procedures, and you can go for the summer training program. Just you take a note of this or take a snapshot of this, that will be helpful for you. And again, these are the programs that you can able to benefit, that is benefit for the undergraduation and the post-graduation students, and also for the PhD students too. They are allowing the PhD students for 2 or 3 months program over there for understanding the techniques, how the lab works, and what are all the experiments that they are going on. Once you get into this website and know all the details, you'll be, all these things has been mentioned very clearly in that website. You can able to know that. And these are some kind of funding for this undergraduation, post-graduation students, and also the PhD students. And those who get a PhD gold medal in post-graduation, they are eligible to get a DST Inspire scholarship. I don't know whether you are aware of this. Aware of this Inspire program. So everyone should be aware of this Inspire program, and there are many Indira Gandhi scholarship for single girl child and postgraduate merit scholarship for university rank holders. And if suppose you are undergraduate gold medalist or undergraduate rank holder, you can apply in UGC website, that is University Grants Commission. You have a scholar, you will be getting a scholarship of 4,000 or 5,000, if I'm not wrong, monthly, I think so. So you can get scholarship like that. Seek help from your guide or the professors over there, and you can start doing that. You should have what kind of funding agencies that's been going there: the DST, DBT, ICMR, and UGC. UGC provides many fellowship. You should track them. You should track them regularly. And there are women scientist scheme for PhD scholars, and those who are about to finish the post graduation. Just you have the knowledge, and those who are going to get gold medal in your university. Make sure that you apply for the DST Inspired Scholarship. And when you are doing PhD, you will be getting an enormous amount of, if I'm not wrong, 25,000 to 30,000. In nowadays, like we used to get only 10,000, 12,000, 8,000. This is a very minimal amount, but now the funding has been increased a very lot. Like the central government has been increased these kind of amounts. So you can make use of this. You'll be having a contingency amount. So everything will be there. So make use of this websites. It helps in giving the scholarship things. And that's all about my lecture. And I would like to thank my guide, Dr. K. Sasikala. She's an eminent professor in human cytogenetics, and she's a retired professor. And I would like to thank her at this moment, because the place where I am, the position, whatever I am, I am my whole gratitude to her, because all the knowledge and all the research things that's been taught by her, by her only, I'm being here. So I should thank her, and also I'd like to thank Professor Vijay Anand sir. He's a professor and head in Human Molecular Genetics Department, and he is the one who asked me to give the invited lecture over here with the help of Purni ma'am. So, and also he's a role model for me for all these things. He is a very humble and polite person. So if you go to Bharatiya University, you can meet him, and he'll give many suggestions. And there is a Department of Human Genetics and Molecular Biology for master graduate students, and also the PhD students also. Okay, so this is my professor. He's a present. He's a president of our institute. His name is Raman Kakabelos. He's a renowned neurosurgeon and neuroscientist, and he has received many awards. To his credit, he has many inventions. He is a pioneer in Alzheimer's and Parkinson's disease. He is treating Parkinson's disease without any drugs. We are doing by pharmaceutical products, like the pharmaceutical biotechnology products. There is no any chemicals in that. So he's pioneer in that. So, and these are the lab pictures. Like this is the NGS that's been newly installed. We are working on this. This is how this next generation sequencing looks like. This is the updated one. And this is my colleague. He's a epigenetist, and he's working in NIH, National Institute of Health. We have collaboration with him, and he's a medical biotechnologist. And this is my team members. And he's an EEG person who works with the brain mapping, and they are the biochemist. And this is my complete molecular biology lab. This is how we work. This is an open array. I was explaining about the pharmacogenetics to you, and this is how it looks. The pharmacogenetic, the pattern looks like. And this is my molecular biology table. This is a biochemistry lab where I used to work, learn the biochemistry techniques. We have our own biochemistry lab to perform for the dementia patients. And these are my team members who are associated with my research almost for 12 years. And he's an ICGB scientist in Delhi. And he's an assistant professor in Korea in Sejong University. He's also an assistant professor in Korea, Dankook University. And she's a researcher in Perugia University, which is in Italy. These are my team members who's associated with me. And that's all. I'd like to finish my lecture and see this is how a scientist or a student perception should be. See here, you could see this slide looks like a frog, if I'm not wrong, and actually it looks like a horse. See, when you tilt the structure, it looks like hours. So this is how the mind of a young scientist or a budding scientist thinking should be like that. You should see in a way where others should not see. You should perceive in a way like where others could not able to see that. So that's all about my lecture, and thanks for your time. Due to restriction of time, I couldn't be able to explain more about the techniques and other things. In future, if I could able to present a, detailed lecture about the techniques, I will perform that time and I will show it to you. And thanks for your time. And I'd like to thank Purni ma'am, and I'd like to thank the students, because I know that sitting for 1 hour is a tedious job. I know, because I'm a neurogeneticist, and I know that because the human brain works for only 20 to 30 minutes consciously. But thanks for your time, and thanks for the opportunity, ma'am. Thank you. Thank you. And if you have any doubts, please feel free to ask me. Any doubts are welcome. Thank you.
Thank you so much, sir, for your thought-provoking talk. Now it's time for feedback session. Students are requested to clear their queries. Students, you are requested to clear your queries.
Arun sir?
Yes, ma'am.
Are you now working for this mutated version of this COVID virus?
A part of the project work is going on. Yes, ma'am. Yes, ma'am. A part of the project work is going on, because like when we put vaccine, like there is a vaccine where we are using is AstraZeneca. So we are like in some patients there is this coagulation takes place. So we are identifying that, ma'am. We are working on that particular project. There were no such rumors behind that, because I encourage to put the vaccines, ma'am, because like generally vaccines will have side effects. The vaccines have side effects, then our body is responding to that vaccine. If we are having fever, if we are having anything, my personal suggestion is like the vaccine is working. And presently we are working on this vaccine project, ma'am, like because there were many rumors that when we put vaccines there will be a heart attack and there will be some blood coagulation is happening. There are many rumors behind that, ma'am, but so to sort it out, to make it very clear, we are working on that.
Okay, sir.
Thank you.
Now I hand over the session to the vote of thanks, Mrs. Aisha.
Yes, ma'am. Am I audible, ma'am?
Yeah.
Is the video clear, ma'am? Can I proceed?
Yeah.
Yes, ma'am. Good afternoon, one and all. It's indeed a great privilege in proposing vote of thanks at this August virtual gathering of Second International Conference on Sustainable Development Goals in Bioscience, jointly organized by the Bioscience Departments of Vivekananda College of Arts and Sciences for Women, Autonomous, LIM Palayam, and Vivekananda Arts and Science College for Women, Veerachipalayam. At the outset, I bow my head to the Almighty for bestowing upon us this wonderful day. I am humbled to express my sincere gratitude to our Honorable Chairman and Secretary, sir, for giving us all the facilities, both online and offline, to conduct events like this towards the upliftment of the students and the society. Thank you, sir. My hearty thanks to our beloved Managing Director, Madam, Joint Managing Director, sir, Joint Secretary, Madam, Vice Chairman, sir, and Director, Madam, for their blessings and support. Thank you, ma'am. Thank you, sir. I extend my sincere thanks to our dynamic Executive Director, sir, for his guidance and motivation in conducting this conference. Thank you, sir. My special thanks to our dignified Chief Executive, sir, Admission Director, sir, and Director Academic, sir, for being the driving force in all that we carry out. Thank you, sirs. I am pleased to record my sincere gratitude To our dynamic resource person of first technical session, Dr. Arun Mayalagan sir, for unleashing human molecular genetics, cytogenetics in an interesting way. Chromosomal studies and chromosomal aberrations has always been an area of enormous discussion in the field of science. Thank you, sir, for throwing light on this area. I am bound to thank our respected Dean of Biosciences, Dr. T. Shankar, sir, for being the mastermind behind this conference. Thank you, sir. I am at the lack of words in conveying my heartfelt gratitude to our respected heads of biochemistry, biotechnology, botany, microbiology, nutrition and dietetics, and zoology of both Vikas and Vyas for their untiring efforts in meticulous planning and executing this conference. Thank you, ma'am. Thank you, sir. I convey my sincere gratitude to the faculty members of bioscience departments of both Vikas and Vyas for their hard work and dedication in conducting this virtual conference. Thank you, ma'am. Thank you, sir. I am humbled to thank the active participants from various colleges of various states and countries for their virtual presence. Thank you, everyone. An event like this does not happen overnight. The wheels start rolling days ago. I express my heartfelt thanks to the technical team for the uninterrupted session and for the YouTube live streaming. I convey my sincere thanks to the students of bioscience departments of both Vikas and Vyas for their enthusiastic participation today. I convey my sincere thanks to each and everyone for their contribution in conducting the first technical session successfully today. I sincerely hope and eagerly await furthermore interesting and informative technical sessions ahead. I thank for the opportunity given. Thank you, everyone.
Aisha. Dr. Arun, sir?
Yes, ma'am. Yes, ma'am. I'm here, ma'am. This time.
Yeah, we have a few questions also
Yes, ma'am. Yes,
in live chat.
yes, yes, ma'am.
We will share the screen of that question. Just a second.
Yes, ma'am.
Is it on you? Pavitra Karthikeyan, you have a question.
Okay, ma'am. Like, yeah, I got that question. Like, Pavitra Karthikeyan has asked me, like, if grandfather have genetical disorders and my mother is carrier, why it affects me with the disease? Yeah. Yeah, the thing is that, like, yeah, as I was saying that if you have clear explanation about that Punnett square, like due to this recessive inheritance, there are chances. As I was saying, it is completely depends upon the luck. How this fertilization takes place, there is a chance of getting a disease, because like you are a carrier of your father is a carrier of your grandfather. So if there are chances, if it is a normal child, then no problem. Being in recessive inheritance, only 25% of like the offspring will be like the normal offspring will be 25%, whereas the 50% we have a chance of a carrier and 25% will be the affected. So the best thing is like that you have to go for a genetic counseling. If you are having any suspicion like that, you have to go for a genetic counseling. You have to go, and then if you go for the genetic counseling, then when you are getting conceived, then you have to take a proper genetic diagnosis, whether your baby is having any genetic disorder or not. You have to screen completely. So that's what, see, like I don't have any genetic disorder in my family hereditary. So I don't check any disease, like I know that. So in case of, like, suppose think like my grandfather is having some cancer or anything, I will perform some genetic studies, because like I'm a recessive carrier of a cancer grandfather. So if there are any chances like that, you have to go for a genetic counseling. Now we don't have any awareness of genetic counseling or anything like that. Generally, what we do is like we go for that, go and meet a gynecologist, and they will say like you go and do this normal genetic test, and they don't perform any genetic counseling when we are doing. But generally, we have to perform a genetic counseling. To my knowledge, we have to perform a genetic counseling. How your forefathers, how your grandfather, and how is your uncle and auntie and everything, all the things that is being connected with your father and mother, we should take a genetic tree, and we have to check how far there is a chance of getting a carrier genetic disorder in your family. So we have to be clear enough in that. So this has happened. This is clear explanation about the Punnett square. If you have a clear explanation about the Punnett square, then you can able to understand. As I was showing the slide, no, the recessive inheritance. That is the reason behind that. There is a chance of 50% you being a carrier, or 25% you can be a normal child, or 25% you they will be completely affected. So that's the explanation, I think so. Like if you are satisfied with that, then okay, or else I can explain more.
Okay, Arun sir. Fine.
Fine.
Once again.
Yes, ma'am.
Same Pavitra is having another question.
Yes, ma'am.
Continue the first question.
Yes,
Whether her mother also will be affected, like that she asked. Once again.
No, the mother will not be affected at any case.
What's the cytogenetics place in my mother and me?
See, you may have a chance, but your mother doesn't have a chance. If your mother is a carrier, then you have a high chance being a carrier or being a affected one. But if your mother hereditary doesn't have any carrier thing, there is no at any way like your mother will not be affected. If your father is affected, your father is a carrier. If your mother is affected, your mother is a carrier. That may affect you. If both mother and father is a carrier, then it comes under dominant resistance. So there is a high risk of getting a carrier offspring in your case. Not in your case, in the common case.
Yes, sir. Fine. Time. But yeah, once again.
Yes.
How the COVID of
yes,
vaccine of COVID-19 is effective?
I'm like first they should have a clear explanation how the double and triple mutate COVID-19 has been erased. The main reason behind that is like, see, it's been transformed from one country to another country. That's how this double mutant has been erased. See, this is the see, when a China person goes to US and he has been affected. There, the genetic makeup is completely different from a China person, and he has a different mutation thing. And when this US person goes to the UK, then there will be a different genetic makeup. So this is a combination of 2 or 3 genetic makeup that has been mutated. There is a chance of high number of mutated COVID-19 virus also, but it all depends upon the mutation. That's what generally we are saying, that the foreigners are not allowed inside. Even here also the foreigners are not allowed inside. That is the main reason, because they are transmitting the virus, and the virus which they are transmitting to us will be in a different genetic makeup. When it gets enters inside our body, I will be transmitting in a different way. My genetic makeup will be different, and I will be transmitting in a different way. That's how this double and triple mutated COVID-19 works. But the vaccine for the double and triple mutated, I think so, it won't work. There is no vaccine for the double and triple mutated COVID-19, but it plays a little role. My suggestion is that, like, if you put vaccine, you are 50% safe. I suggest like 50% safe. I can't say that you are 100% safe, but many says that if you put Pfizer vaccine, it is 95% safe. And if you use Moderna vaccine, you use COVID Shield, Covaxin. See, all the vaccine has the same thing. Only the makeup is different. Some is using mRNA, some is using adenovirus, and some they are using this the viral particles. Only the makeup is different, but the concept and other things are everything is same. And I don't think so this vaccine will be helpful in controlling this double and triple mutated COVID-19 vaccine, because it is a highly mutated COVID-19 thing. So it all depends upon the immune response that we are having. So the main thing is like we should have a good immune response. That's the main thing. And I could see one more question is like satellite chromosomes. In that is, yeah, if you have any satellite chromosomes, your chance of getting a cancer. Your chance of getting a cancer. Your P53 gene mutation, there will be a chance, there will be high chance of P53 gene mutation.
One second, Arun, we are checking
Yes,
again.
Yes, ma'am. No problem. Take it.
Okay, that's all. I think the question section is over. And really, I thank you and Vijayanand sir for giving us a good chance to hear about the cytogenetics. Arun, thank you so much.
Thank you, ma'am. Thank you.
So I hereby inform that the next session is starting. All of you, let us assemble by 1:15 because the session starts by 1:30. 1:30 we have the session for the Boston. Sun University, China. Thank you.